All right, welcome ladies and gentlemen to the long-awaited cell transport video. Yes, I know it's not particularly eagerly awaited, and I know that this topic may have been quite difficult for some of y'all in freshman biology, but let us try our best to get through it, and hopefully it will be a little easier to understand this time around. So, I realized that I wanted to go over a few terms and a few concepts with you guys before we went into more depth into cell transport. So I added a few things after your notes were printed. Yes, you may have to write some things down. You can either write on whatever empty space there is on your notes. For example, I know that there's at least a half page empty, or you can write this in your notebook if you'd like. So, I wanted to define a few things and go over some terminology and how you're supposed to phrase things. So, a membrane can be permeable, meaning it can allow certain substances to pass through it. Now, for a cell, for various parts of a cell, it wouldn't make sense to be permeable to everything. A cell has to act as a boundary to keep in certain substances and to keep out other substances. So no membrane for a cell is completely permeable to everything. Speaking of which, when we talk about permeability, we don't say that a substance is permeable. It's the membrane that is permeable or impermeable to a molecule or a substance. If a membrane is impermeable to something, then it will not allow a substance to pass through. So most membranes, especially those in our cells, are actually semi-permeable. They will allow some substances to pass through and not others. And it's not like this is something conscious that the cell is controlling. It's the properties of the cell membrane itself that prevent some things from passing and allow other things to pass. So let's look at an example here. So what we have here is a labeled kind of very quick diagram of a cell membrane. The most basic cell membrane is a phospholipid bilayer, and we're looking at a plain one with nothing else stuck in it. Now, this cell membrane has been labeled. The areas that are hydrophobic have been highlighted in green, and the areas that are hydrophilic have been highlighted in blue. Remember that the heads of phospholipids, which are the nice balls with legs that are sitting there, the heads of the phospholipids are hydrophilic, meaning that they are polar. They are attracted to water. And that's why they face the area outside of the cell. So for example, oops, my bad. This area out here is a watery environment, and this area over here is a watery environment. The interior of the phospholipid bilayer, where the tails are located, is hydrophobic because it is nonpolar, and water will only associate with polar or charged objects, so it will not interact with those nonpolar areas. That's actually what you saw a second ago when the animation accidentally started. So we see various molecules here. At the top we have water. Below that, the two purple spheres stuck together are an O2 molecule. The red hexagon is a glucose, and the tiny little yellowish gold sphere is a hydrogen ion. I know it says H in the key, but that's because I wanted it to apply to the hydrogens in water as well as the hydrogen ion. So let's look at what happens when water attempts to cross the lipid bilayer. Notice that it is able to associate with the phospholipid heads, the polar parts of it, but it is repelled or it is not attracted to the hydrophobic parts. So it almost bounces off of the cell membrane. Let's look at the second molecule now. Oxygen is small. It's uncharged, therefore it is able to pass through the cell membrane fairly easily. This is a good thing because oxygen is needed in a constant supply in our cells, and it would be a little problematic if it needed a transport molecule or some other help to get it into the cell. Now let's look at glucose trying to pass. Notice that while it approaches the membrane, it is too large to fit in the gaps between the phospholipids. Thus, size is a barrier to molecules passing the phospholipid bilayer as well. And then finally, we have a hydrogen ion, an H+, and let's look what happens when it attempts to cross the membrane. So, it is able to interact easily with the polar heads. However, it is blocked by the hydrophobic tails on the inside. So the membrane is permeable to some molecules like oxygen, but it is impermeable to others like glucose, water, and hydrogen ions. However, let's take a look at this new setup. We now have a transport protein embedded in the membrane. And this transport protein will only allow certain molecules to enter. It happens to be a glucose transporter, which we did see in the example of the endomembrane system worksheet. So this is a glucose transporter. It has chemical properties that will only allow glucose to pass, not any of the other molecules. So when glucose comes near it, it can actually pass through and emerge on the other side. This isn't done using any energy by the cell. In fact, the cell doesn't expend any energy beyond making the glucose transporter and sticking it in the membrane. What happens is the glucose molecule fits through that channel so it can flow into or out of the cell. So channel proteins, when they're embedded in a cell membrane, can change the permeability of a membrane. In this way, you can also have a cell control the permeability of the membranes that are found in different areas, whether it's the plasma membrane at the boundaries or the membrane that surrounds a particular organelle. Just by putting different membranes into that protein, it can change what can pass easily in and out of that membrane. So let's get back to the notes. We need to clarify a few terms before we move on. Hopefully you remember this from your basic chemistry classes, but in solutions, there is always a solute and a solvent. Solutes are molecules, sometimes small solid particles, sometimes other liquids that are dissolved into another liquid or fluid. The solvent is the fluid that is dissolving the solute. In our cases, since we're looking at biological systems and all of our biological systems are basically made up of what are called aqueous solutions. An aqueous solution is watery. We'll say water-based. So our cells are aqueous solutions, meaning they're water with a bunch of stuff dissolved in them. The environment outside of our cells is also an aqueous solution, water with proteins and other molecules dissolved in it. So in our cases, when we're talking in biology, our solvent is almost always going to be water. So let's talk about different types of transport. Our first is our lazy guy just lying on his back with his leg up in the air for some reason, just kind of hanging out, chilling. He's supposed to represent passive transport. He is passively just kind of lying there, not doing anything. So passive transport doesn't really require any effort from the cell. You can almost think of it, if you'd like, as lazy transport. It's lazy because it doesn't require any energy. Notice that I have here, it doesn't require any metabolic energy. That's just the chemical energy that your cell uses, meaning ATP. And one of the more famous examples of passive transport is diffusion. You can't think of them quite interchangeably because diffusion is a type of passive transport, and there are two other types. So diffusion is the net movement of molecules from a high concentration to a low concentration. Another way you can phrase this is that molecules are moving down or with the concentration gradient. A concentration gradient is just a difference in concentration across a distance. So what we see here in, let's say, panel number 1 is a concentration gradient. You have two solutions separated by a lipid bilayer, one of which contains large amounts of these hexagon-shaped molecules, the other of which contains none. So we have a clear difference in concentration here. What happens is, as long as molecules are able to pass this barrier or pass through space, they will move until they basically reach equal concentrations. This is not a conscious decision on the part of the molecule itself or of a cell. What happens is all molecules are, unless they're at absolute zero, have some motion. They have energy which causes them to move around. So they're going to basically float through space bouncing off of things. You can think of them like a bunch of ping pong balls dropped into a box. They're going to bounce around and hit each other and knock each other all over the place. When this happens, they'll tend to be bounced around, and eventually they'll bounce to where there's areas where there's fewer of them. It's the same way in which if I packed a bunch of you into one corner of the classroom and then said, okay, you're free to move about as much as possible, you would probably start to spread out. Or, like I said, if you dropped a bunch of ping pong balls into a clear box and were able to look at the movement of the ping pong balls, as long as they had energy, they would bounce around and hit each other and spread out until they almost evenly filled up the space. So molecules will move, they'll bounce into each other, and they'll tend to spread out. Eventually, they'll reach something called equilibrium. And if we look at the third panel here, this system has reached equilibrium, by which I mean that it has equal concentrations of this molecule spread throughout. There is no longer a concentration gradient. But I want to clarify something a lot of people kind of confuse. While the system is at equilibrium, it is not unchanged. So here's what happens. Remember, all molecules, unless they're at absolute zero, are in constant motion. That means these molecules aren't going to stay still. This is a system of inanimate molecules. None of them are thinking, oh, I've got to spread out because that is what I as a molecule do. Instead, they're still bouncing around. So what will happen is some of these molecules may cross the barrier, but on average, for every one that crosses in one direction, another one will cross in the opposite direction. So at any given moment, if you measure the concentrations of this hexagonal-shaped molecule, it will be the same on both sides of the barrier. So what we say is that there is no net movement. What that means is there is no change in the total concentrations. And like I said, for every molecule that crosses in one direction, another one will cross on the other side, and the concentrations will even out. And it's not that for every molecule that crosses to, say, the top part of this diagram, another one is forced to cross down to the bottom. It's just as they move and bounce into each other, they'll still spread out. And so on average, one will cross in either direction. Apologies, apparently my computer has decided to give you random warnings. All right, so there are 3 different types of passive transport. I know 2 are listed here, but that's because the third one is kind of a subset of the other 2. So these 2 types of passive transport are differentiated by whether or not the membrane is permeable to these molecules. What that means is, if a membrane is permeable to a molecule, it can move through simple diffusion. That's stuff like CO2, O2, small uncharged gases, as well as very small lipids. Basically, if it's small and uncharged, then it can likely pass through and move by simple diffusion. However, there are a lot of molecules and other particles that you would like to have easily move across the cell membrane or a membrane inside of a cell, like glucose, water, various ions that cannot pass through the cell membrane unaided. But you still need to transport them easily, and a lot of times it's still easier to transport these things by diffusion. So instead, you have these transmembrane proteins. They act like portals or gateways that are very specific to certain molecules and will allow them to cross freely. And a lot of times these transmembrane proteins will not be directional. They'll just be these open pathways that molecules can pass through in either direction. And we'll talk about some more of those as we go through. So here is a glucose transporter. It can transport glucose down its concentration gradient without the use of energy on the cell's part. So here's how this happens. In step one, what we see is an empty glucose receptor, a glucose transporter that has a glucose molecule come in from outside of the cell and bind to it. And that's what we see right here. Then what happens is that binding of glucose actually spontaneously changes the shape of that molecule, and now the protein, instead of opening to the outside of the cell, opens to the inside of the cell, which is the step that you see here. That conformational change actually allows the glucose to be less attracted to the interior of that protein, and it can pop off towards the interior of the cell, which is what you see happening here. Once the glucose is released, that glucose transporter is no longer held into this other open shape, and it will then split. It will then change shape spontaneously to go back to its original shape, like what you see here, where it is now free for another glucose to bind and continue the same process. So this thing can just continuously transport glucose molecules into the cell. It's the binding of the glucose that changes the shape of this protein, not the addition of any energy on the cell's part, so the cell doesn't have to do any work to move the glucose around. Osmosis is the third case of passive transport that I was talking about. It is simply the diffusion of water. So molecules will move until an equilibrium is reached, but sometimes the solute molecules cannot move. Instead, you have a semi-permeable membrane that will allow water to move, but doesn't allow the solute to move. And when this happens, then water will diffuse across that barrier until the concentrations are basically equalized. We would try and do an experiment like this in class, but unfortunately, you have to have basically some sort of membrane that you can very, very strongly adhere to the sides of a container, and I don't think we have something like that easily. We will try and do some experiments in class with something called dialysis tubing, which is semi-permeable, so you can see some examples of osmosis firsthand. However, we're not going to be able to do this experiment right here. All right. There is an entire section of your notes here that I decided to save for a different lesson because it's kind of the complicated stuff, and I want you guys to get what we're talking about here first. So I decided that this is going to be its own separate lesson later. So for now, I'd like you to skip and go down to where it says facilitated diffusion. Facilitated diffusion is, like I said, another type of passive transport. It occurs when you have molecules that cannot pass through the cell membrane. So for example, this red molecule right here can't pass through. This blue molecule right here can't pass through, whether it's because of charge or whether it's because of size. Then you can, if you need these molecules to be able to pass through the cell membrane into and out of the cell easily, the cell can insert membrane proteins into it to allow these molecules to pass through. There are two types, a channel protein and a carrier protein. A channel protein essentially has a hole through the center, a channel, and it will allow molecules to pass in and out. A carrier protein will physically transport a molecule across the membrane. There isn't really a lot that you'll have to use to differentiate between these two. Honestly, you mostly need to know that they move molecules across the cell membrane without requiring energy. So now we move on to the second type of transport. Active transport. As you can see, our previously lazy guy has stood up and is now running. He's actively moving from one place to another, and that sort of movement requires energy. That is because active transport is actively done by the cell. Now, this is the cell actively trying to move things. This sort of movement does not happen spontaneously. It requires the use of energy and effort on the part of the cell. It is going to move molecules in the opposite direction that passive transport would. So it's going to move molecules from a low to a high concentration, or you can also phrase it that it's going to move it against the concentration gradient. This is usually done when you are trying to build up a concentration of some molecule or ion, and that's going to be a concept that we're going to come across repeatedly, especially when we talk about photosynthesis and cell respiration and the transmission of basically signals in nervous cells. This often uses transmembrane proteins, but we're also going to talk about a type of active transport that's not using proteins embedded in the cell membrane. So pumps are going to be the most common type of transmembrane protein that's used. It's going to actively move molecules across the membrane, and there are two very important examples of these. The first, which you probably heard about a little when you were in freshman biology, is the sodium-potassium pump. What it does is it basically pumps sodium into the cell, and it pumps sodium to the outside of the cell, and it pumps potassium to the inside of the cell. Sorry, I got those captions wrong. So switch this and this with each other. The sodium-potassium pump works to build up a concentration gradient with more sodium outside the cell and more potassium inside the cell. And we'll talk about this more when we talk about neurons. The other type of pump is more often used in photosynthesis and cell respiration, and it's used to build up a concentration gradient of hydrogen ions. This is called a proton pump because if you think about it, a hydrogen is just a proton circled by a single electron, but if you take away that electron and it becomes a hydrogen ion, it is basically a naked proton with nothing else around it. So you can also call hydrogen ions protons. A proton pump will pump those hydrogens from one side of a membrane to another. And this doesn't just happen from like a cytoplasmic area to an extracellular area outside of the cell. This also happens within the cell. So for example, mitochondria have a lot of proton pumps in their membrane that build up a concentration gradient, as do chloroplasts. Now, there are also other types of transport, for example, bulk transport. Bulk transport, just like you buy in bulk someplace like Costco, bulk transport means you're moving a lot of stuff at a time. And this involves infolding and outfoldings of the membrane. The first one we're going to talk about is endocytosis. If you remember terms from way back in the day in maybe life science class, you may remember hearing about an endoskeleton. Endo means inside. Cyto means cell. So endocytosis is the process of bringing materials inside the cell. Remember when I said you may have heard about an endoskeleton? Well, an endoskeleton is the type of skeleton we have that is internal to our body. Endocytosis is the process of bringing stuff to the inside of the cell. So there are three major types. Basically what ends up happening in any form of endocytosis is the cell membrane acts almost like you're pushing a finger into it. It forms this depression that pinches off and forms a vesicle that moves to the inside of the cell. It looks very similar to what happens with the Golgi when there are vesicles butting off of it to move around the cell. A specific type of endocytosis is phagocytosis. Phage literally means to eat. And so this literally means cell eating. This is because this is a process that single-celled creatures use to eat solid particles or even other single-celled creatures. It is used in our cells. For example, white blood cells take in bacteria and pathogens and disease-causing agents. By endocytosis, take them in and then usually break them down and digest them. There is also penocytosis. Literally, peno means drinking. So this means cell drinking, and that is because you're not taking in solid particles. Instead, you're taking in some of the solution surrounding the cell or the extracellular fluid, and so it's almost like the cell is drinking in small chunks of the fluid around it. Once again, you can see this kind of budding where basically there's an infolding of the membrane. It pinches off here and then forms a vesicle that can then move into the cell. And the last type of endocytosis is receptor mediated. A receptor is something that receives a signal. So you'll have these patches of receptors on the surface of a cell, and when they bind to a specific molecule, which is this little green dot right here, that will trigger a reaction that will cause them to form a vesicle that will then bud off of the cell membrane and go to the inside of the cell. This specific molecule can be anything. It just has to be something that interacts with that receptor. So this will be different depending on the cell you're looking at. Usually this occurs at something called a clathrin-coated pit. Clathrin is kind of this hairy looking stuff that will stick to a vesicle, and it's important that this happens at those locations just because clathrin is a molecule that helps form these kind of bulges in the membrane and helps it pinch off to form vesicles. So that is the end of what we're talking about here. You can now go on to do your homework. And yes, I'm sorry, homework sucks, but it is necessary to help you guys learn.